Hello there, my name is Riddhi Singhvi, and the title of my project is CRISPRing Up the Genes, A Tale of Two Experiments. Here's my table of contents, here's my abstract, and here's my background. CRISPR stands for Clustered or Regularly Interspaced Short Palindromic Repeats, and for my project I use CRISPR-Cas9. Cas9 is a CRISPR-related enzyme which acts as a molecular scissor, but all scissors need a sense of direction. That's where guide RNA comes in, also known as gRNA. It's a type of RNA molecule that binds to the Cas9 and specifies, using the sequence of the gRNA, the exact location over the Cas9 will cut the DNA. DNA is a molecule that almost all organisms use to store genetic information, and RNA is a molecule corresponding with DNA that living things use for a multitude of reasons, including transporting and reading the DNA instructions. There are three main genetic edits that can be performed with CRISPR-Cas9. The first of which is disrupt. It inactivates the gene. The second is delete, which deletes the intervening sequence. And this third one is correct or insert. With the use of a DNA template alongside the CRISPR-Cas9 machinery, you can allow the cell to correct a gene or even insert a new gene. In my project, there were two major edits. The first of which is streptomycin resistance. It's a type of antibiotic that binds the ribosome and prevents it from making protein, leading to the bacteria stopping its replication and growth. The DNA modification in my project changes a single DNA base, so the lysine amino acid at position 43, also known as K43, is turned into threonine, which is also known as T43. The second edit was fluorescence. In this project, there is the use of a green fluorescent protein, also known as GFP. It's a protein that exhibits a bright green glow when it is exposed to light from the blue to ultraviolet range, and it is oftentimes used in biotechnology to mark specific cells, or as a reporter gene which shows if a cell expresses a certain protein. In this experiment, there is the transformation of a plasmid that contains a GFP gene into E. coli cells, and the transformation conditions will be varied during the experiment to determine how it changes the cell's ability to take up the GFP plasmid. The fluorescence experiment was also done by Jennifer Doudna and Emily Charpentier in 2020, and they won the Nobel Prize for CRISPR-Cas9 genomic editing. The question that I posed during my research was, is it possible to investigate the relevance of a DNA template using CRISPR to edit the genomic data of E. coli to make it streptomycin resistant and fluorescent? I wanted to see when performing two different DNA edits simultaneously, were they mutually exclusive or would they affect each other? Here's my hypothesis. E. coli bacteria resistant to streptomycin can be created. The relevance of a DNA template can be investigated when using CRISPR to edit the genomic data of E. coli to simultaneously make it streptomycin resistant and fluorescent. Editing two genes at the same time will not negatively impact the growth of E. coli on three different types of agar mediums. Here are my materials and equipment. For my project, I use perishables, non-perishables, and here are my instruments. Here's the visualization of the edited streptomycin gene. There's the target, rank, and target sequence. And here's my procedure. For my project, I used three different types of agars, and I used three different solutions. I used a standard CRISPR mix, and with 50 microliters of the GFP plasmid, and a standard CRISPR mix without a template DNA. For my project, I also used two different types of incubators, a 30°C incubator and a 37°C incubator. The 37°C incubator was used for the E. coli, and the 30°C incubator was used for the CRISPR. The reason why I used a 30°C incubator was because the CRISPR-Cas9 needs time to work, but if you put it at such a high temperature so quickly, the CRISPR-Cas9 will be rendered useless. I also used a 42°C heat shock, and I followed all safety guidelines that were necessary for a project of this nature. Here are my variables. I have my control variables, which was type of agar, temperature, amount of liquid used, time, CRISPR recovery time, strain of E. coli, phosphine glycol, and CaCl2, also known as calcium chloride 2. Phosphine glycol and CaCl2 were used for my project because both the CRISPR-Cas9 machinery and the E. coli cell walls are negative, which they repel each other. So the phosphine glycol and CaCl2 is used to coat the CRISPR-Cas9 machinery and neutralize the negative charge of it, so it has an easier time inserting into the E. coli cell walls. My independent variables were type of agar, plating, gRNA, template DNA. And my dependent variables were bacterial growth, amount of E. coli produced, and type of E. coli produced. During the experiment, some qualitative details that were observed were some of the plates had uneven agar distribution, which led to some of the earlier plates having more agar and the later plates being too thin. And this was really evident when doing some of the experiments and seeing that some of the plates barely were able to grow any bacteria due to such a light film of agar. The growth of cells over the 2-day period in the incubator. And when scraping off the bacteria with an inoculation loop, it was extremely surprising to see how different colonies could look even though they were the same bacteria. Here are some potential sources of error during my experiment. There was some background contamination of microbes. Some of the plain agar plates had some background contamination from outside sources. There was no way to ensure that there was uniform spreading of edited bacteria on agar surfaces, and when working with such small quantities of materials, such as 10 microliters or less, can introduce measurement errors, even though all pipetting was done with a mechanical pipette and utmost caution. Here is my Table 1 results. Here is the unedited E. coli, the CRISPR E. coli with the GFP plasmid, and the E. coli without DNA modifications, and my 3 different types. Here is the knockout efficiency. As you can see, it was all 100%. Here is my first graph. It shows the growth of E. coli in different agars. So, as you can see by taking a look at the graph here, the unedited E. coli and the E. coli without DNA modifications did not survive on the strep/con agar plate. This is because E. coli isn't naturally resistant to streptomycin. But for the CRISPR E. coli and the CRISPR E. coli with the fluorescent GFP plasmid, they did survive on all 3 types of agar. But let's take a look at that graph from a different perspective. As you can see, it's a little more evident here that the unedited E. coli and the E. coli with the DNA modification did not survive on the strep/con agar plate, but they did survive on the LB agar plate and the conamycin agar plate. Here are the images of my project. On the left, I am preparing the CRISPR-Cas9, and on the right is the centrifuge that was used in testing. On the left is the CRISPR-edited E. coli under LED light, and on the right is the CRISPR-edited E. coli under UVA light. Here on the left are the plates in the incubator, which this was the 30 degrees Celsius incubator, and on the right are the 2 E. coli strains set with 1 agar type. On the left is the base E. coli bacteria plate used for testing, and the E. coli bacteria on streptomycin agar under UVA light on the right. Now, let's take a look at the results of this experiment. The E. coli was made streptomycin resistant and fluorescent using CRISPR gene editing, and it is possible to do this by editing 2 separate genes. The simultaneous editing of 2 genes was not different than the separate editing of the genes. The edited genes were mutually exclusive, and they did not affect each other's properties, and there was no negative impact of editing these 2 genes simultaneously. Now, I'm happy to tell you that in conclusion, the research problem was solved. The experiment to edit 2 genes at the same time with different DNA templates was successful, as the 2 edits did not interfere with each other. And in the end, the results were E. coli cells that were fluorescent and resistant to streptomycin. Although the E. coli bacteria grew robustly even with extremely small quantities of culture. And this experiment can be extended to other properties, such as using the site ChopChop, which I used for the graph shown earlier, and selecting properties and genes to modify. You can design your own gRNA using this site, and you can select whatever you want to modify, and you can perform all 3 of, experiments such as deletion or correction with ChopChop. A multi-gene edit would be crucial for future experiments in biotechnology, especially in medical treatment. Currently, CRISPR technology is used for diseases that are caused by one flaw in a single gene, such as sickle cell disease, but by using multi-gene edits, it can spread out to a much larger field of study. Here's my bibliography. Thank you for your time.